So our final speaker today is going to give you some very positive information, which may sound negative, but it's really very positive information about the research that he's doing at Duke University with Dr. Neil Spector. Dr. Tim Haystead is a professor of pharmacology and cancer biology in the cell and molecular biology program at Duke University School of Medicine. Dr. Haystead developed a chemoproteomic drug discovery platform, which he used successfully to identify several small molecules as promising imaging agents for the detection of metastatic cancers. More recently, Dr. Haystead was persuaded by Dr. Neil Spector, whose own 17-year journey with Lyme disease that caused carditis resulted in a heart transplant in 2009, to direct his technology at Lyme disease. This line of research has already identified a novel diagnostic imaging agent for specific Borrelia, as well as multiple novel Borrelia inhibitors. Dr. Haystead.
Well, I was glad to talk to you guys about this stuff. My audience is not normally patients, it's other scientists. So I'll try my best to tell you about what we're doing, and we'll get right in there. So I'm between you and lunch. I know that. So this is my favorite protein in the world, and it's one where we have put 2 drugs in the clinic, and I'll tell you about that in a second. But a special mention to Dr. Neil Spector, who actually introduced me to the Lyme world and asked if the technology we had applied and the way for cancer would have utility in treating this or attacking this organism. And it's been quite a journey, and really enjoyed the interactions. And another key member is Monica Embers, was here yesterday. And we form a nice little team where we think we can get some genuinely new treatments for this disease, not only to attack it, but also to see it. In cancer, we call this a see and cure strategy, because the field that I'm in chemical biology, we make novel molecules that target specific proteins that have been identified associated with disease. And we feel like we're changing some of the paradigms there. And actually, I think the work we're doing in Lyme is going to change some of the paradigms in the way we think about infectious diseases, that maybe we could combine imaging and treatments. So we're going to tell you a little bit about that. This is Neil. He's a very handsome man, and he's not yet. That's Neil. But the point of this slide actually is that there is, between us, I think it's safe to say, Neil's got me beat because he's got 2 in as NDAs, and I'm a little bit behind, but I have 2 drugs that we've developed. These were scratch discovered. They did not exist, using a platform technology. And the first one, we did it in this garage in downtown Durham. Literally, and we made something. I formed it in 2000, and at 2008 it was bought by Pfizer for these pills. And I'm happy to say that even last week, this is been through multiple phase 1 trials for solid tumors. It progressed reasonably well through phase 2, and I heard that it was approved for phase 3 by the FDA a couple of weeks ago. So we're well on the way to making this into real pills to treat people. Obviously, during all of these clinical trials, people have been taking this drug, and I'm happy to say that it has helped people. So at Duke, in collaboration with several investigators, including physicians like Neil and another one called Dr. Liley, we developed another imaging agent based on the same kind of technology where we can image metastatic disease. And there's a clinical trial that started last year on that one, so I'm pretty excited about that. And Neil has put lapatinib, which is a drug that has had great impact with women in breast cancer, and another one that he developed while he was at GSK. He's also been involved in several INDs. The point is that we have a team of people that know how to take novel things and move them forward, and that's our goal here. So this is the organism, and I'm a chemistry lab. We actually grow this in the lab now, because we make things. I like to be able to test them on something. And Monica Embers was very instrumental in teaching us how to do this. So I have students that grow these organisms, so they can test things that we find directly, and then we pass them on to Monica to see if they're actually correct, if they actually do things that we think they do. So that's a bright, what's called a dark field image of the organism. It's very cool. It's got this long spirally Keaty thing. So one of the first projects that we applied to technology, and we called it proteome mining, and we target a particular piece of the genomes in all organisms. We call it the purinome. It's actually things that bind this molecule called ATP, which is the energy in all our cells. But what we had been able to do is we found a way we can catch all of this. So in Borrelia, for example, there are something like 1,300 annotated genes. About 10% of these are enzymes that use this molecule, ATP, and that's a lot of potential drug targets. If it binds ATP, in my mind, it's a potential drug target. And in the Borrelia genome, there's something like 130 of these. In human, there's about 2,000. So a lot of potential. So what we've done is that we applied, and where we are, and this work is funded by the Cohen Foundation, who funded it completely. What we've been able to do is take that 130 targets of the Borrelia genome, get together with Monica and other experts in the field, and define a number of those genes that might be, if you inhibit those enzymes, if you had an inhibitor, it would have an impact on the organism. So our way to pick out of the 130 what would be the ones to really screen and find new things, and we're using the knowledge that others have accumulated as a jumping off point. We have about 20 favorite targets, and these are some of the targets where we have expressed these as recombinant proteins for our assay, and we're taking them down this drug discovery pipeline. We are literally doing this every day with the technology I think I'll describe in a moment. But we express these as recombinant proteins, and each one of these, this is where we are in this pipeline list. Some we've gone quite far. One of them I'm going to talk about, this HTPG target, is preclinical. We're ready to move it into the clinic for imaging and almost certainly treatment. These other ones are novel, exciting targets where we have been successful in expressing them and introducing them into our screen. And by the end of this year, we should have done all of these, and we should be somewhere well down this path. That sounds like very ambitious, but the goal is to identify what we call lead molecules that we can then whittle down and say, well, we can advance 3 of these, but behind this will be a lot of others if these should fail, and work with the Cohens to come up with a strategy to move it from lead to a medchem campaign and into actual preclinical studies. Where we have gone quite far is with this one, and I'm going to talk a bit more about that, where we are now in this preclinical era, and we're looking at cell assays, preclinical animal studies now. Okay? And this all happened within 18 months when we got funded. So, one of the, this is data, sorry, I cannot not show data, okay? So things that have come out of this that have just opened up a new world for me is that this protein, we call it HSP90, that was that big molecule I showed earlier, has a Borrelia equivalent. And the sort of things that we can do with this technology is we take this target, this is the human form, it's called HTPG in this organism. It's also found in other prokaryotes. But actually what we're able to do is express these proteins as recombinant and then introduce them into a quantitative assay. And you see all of these compounds that are listed here. They are different hits or lead molecules that target this enzyme. And we started to look at it against these enzymes. And what we found, to our surprise, so we were very good at medicinal chemistry at making inhibitors for the human form. But what happens if we take those libraries that we had made and spent years on and try them against these? And what we found is that they broke out into specific chemical matter space that are very, very potent inhibitors against the Borrelia version. And some of our more synthetic ones were actually not so great now. But what we did find is a couple of scaffolds that are new that specifically target that organ. So that's pretty exciting to us because enzymatically and against this target, or this target here, we can now advance this. But we also can compare to, we talked about the microbiome. Well, let's introduce that into the equation very early on. Let's try to weed out things that might also hit this. So again, enabling us to be more focused. But the good news of all of these studies was that when we took the cultured organism and we started to test them in our in my lab just next door to some new space we were given, we showed that some of them are indeed active against the organism. So it's a promising start. What's the next step for this? Send them to Monica, try them out, see if they work in her animal models. So some of them don't work, some of them are great. So you can see out of all that list over there, we start to pick candidates that look pretty promising. So it's a very, very quick process. I can tell you that previous screen, it takes us 3 days to go through our library now. It takes a minute per assay to do each one of these screens. So we have massive throughput. So, and we can take things forward. So this is, I want to talk about, and I want to move the topic to the imaging side because I'm just so excited about this because we had, when we were looking at cancer, one of the interesting things about our protein of interest, which is called HSP90, is that when a cell becomes metastatic, it turns out that our favorite protein that we just made drugs to liked to migrate to the surface of metastatic cells and became associated with aggressive disease. And we end up making probes that specifically identify that surface form, and it enables us to image it. And I can show you some of that in a second. But we also had a series of analogs again, based on that program, that didn't hit the human form, but does hit, now we know, the Borrelia form, another imaging agent. So what we did was we made fluorophore versions of this, and the little movie was going a second ago. You could see the tail. This is called a lollipop, and that's one of these bleb-like structures that really absorbs this probe. That's sort of a taste of what's to come. So going back to cancer, how do we look at these things? How do we know that they go where we think they go when we put them in animals and things like that? So the way we've done, we do this, we can go back this several ways, live imaging, which you'll see in a second, to more detailed histological type imaging approaches. So in cancer, what we found is that tumors is a tumor on a mouse. I'll explain what this is in a second. They express this surface form of HSP90. It's associated with metastatic progression. We were funded by the DoD. We had an $18 million grant based on that observation to take one of these things into the clinic to demonstrate that in human beings you could see a similar phenomenon. So in this mouse model, what we do is we inject one of our probes, and it's a probe, in this case called HS131, and it has a fluorescent tag on it that glows red when you excite it. So what they did with this is that they take a mouse and they inject him with our drug and wait 6 hours, and then the mouse is euthanized. And because we're interested in the complete biodistribution of this probe, does it really just go to the cancer? What other cells does it go to? We then cryosection the mouse 700 times, 700 slices of the entire animal, and then image each one with confocal microscopy. So what you see here is a section that's called the bright field. You can see the brain and all the other organs. There's the tumor there. But you look at fluorescence, you just find fluorescence in the tumor. This is all the gut. It gets eliminated very quickly through into the gut. So the majority of the material that's injected in the tail, most of it will be eliminated, but enough of it resides in the serum long enough to find the tumor and just light up the tumor. And if you take the whole animal and reconstruct him in 3D, this is the bright field, all the slices put back together again. You start to look at where else did it go, and you can reconstruct the animal. And what you see is the tumor over here. That's the gallbladder, and you can see how it gets eliminated. There's some sites of natural fluorescence in the eye here, but it's not. This is nothing to do with our drug. It's a compound produced in this gland that the mice secrete. So that's natural fluorescence. So why are we excited about this? We're excited about it because when we inject into an entire animal, it only finds the tumor. And that's actually important because now we can develop agents that specifically target just tumors and not normal tissues. The other finding that's really exciting to us is the path of elimination is in through the gut. And in a single pass, about 90% of it is eliminated, but that is sufficient to find these tumors. It also makes it pretty convenient for the development of a PET agent for human studies. So this is a mouse that has a primary tumor and a secondary metastatic mass. This is FDGT PET in that animal. This work was done at UNC, University of North Carolina. And what they did in this animal is they injected him with FDGT PET, and they took an image. And then the next day, they injected him with a version of our probe that carries radioactive iodine. And what we see is a complete focus of the biodistribution only to the tumor mass, the secondary tumor mass, and the gut. So it's rapidly eliminated. But you notice in the most sensitive way, the probe is not anywhere else. And this is why we got funded to move this into human trials. Because if we could get a similar observation in people, we'd have a diagnostic for metastatic disease. And that's an ongoing trial. So what does that all got to do with Lyme disease, and how can we apply this technology? There are two areas where we're planning on applying this technology on this approach. One is to image T cells during active infection. The other is to image the organism itself in a person with applying the same strategy. Where we are is we've done the inflammation part, and you'll see in a minute we've made a probe that is specific for the organism. So of all the cells in the body, what other types of cells would take up such a probe? In the whole animal studies that we've done, we didn't find any other than tumor cells, but cancer can't have simply just invented a pathway, sophisticated pathway to express this protein on the surface, and it's only associated with metastatic disease. But one of the working hypotheses was that proliferating cells, rapidly proliferating cells, probably express this probe because metastatic cells are rapidly proliferating. So one thought about that was that immune cells, given an immune challenge, you get proliferation of antigen-specific immune cells that proliferate, and T cells are particularly attractive. They split into two types of different populations, and this biology is well understood. But within the days of antigen presentation, you'll see these proliferate in vivo. So we took this probe, which has various iterations, and the idea is that in a resting T cell, resting T cells do not express our protein on the surface because it's not proliferating, they're just benign. But if you activate it, you present it to an antigen, it will express the protein. The protein expressed on the surface will recognize the probe, and we know now that causes the protein to aggregate and be re-internalized and carries the probe in. And because these cells proliferate, that signal gets amplified. And does that work? And you can do all kinds of nice controls. You can compete these things, and we've done all those. So if you look at just PBMCs from people, healthy volunteers, and you make challenges with them, and you look at the total expression of the protein, and the protein is expressed on the surface in T cells isolated under flow and characterize them with their different markers, you see a correlation of probe uptake and activation over time in the test tube. So that's promising. But what happens when you put it in an animal? So where we began this study was in a arthritic model, which is a good one to start, because if it's not going to work in this model, it's never going to work anywhere, right? So happily, there's a number of CROs that we can use that have these models. There's a collagen-induced arthritic model, which is my favorite, and very nice pathology associated with defining that in this animal mold disease. So what happens if we took our probe in this model and then did the slicey mouse? What does the biodistribution look like? So we did slicey mouse, and this is some of the data we've got. We literally got this in the last 3 weeks. The first thing we noticed, if you remember the previous animal, the only place we find the probe is in the tumor. Now when we do close-ups of the entire animal, we've injected him, and you'll see the whole animal in a minute. These are the bright field images. We do see uptake of the probe in the joints in various regions and in the paw regions associated with the pathology of this disease. To me, this is like really fantastic. So this is a good news for arthritis, and if nothing else, it's a great new tool to add to armamentarium for that disease and other autoimmune diseases. But if we rebuild the mouse, here he is. Hopefully, you can see in the whole mouse when he's injected in the tail, the paws lighting up, the joint regions. Here's some poop for anybody that wants to do some fecal transplant. The same sort of elimination through the gut. You can see the site of injection. So we're actually moving towards a PET study with this. So you could apply this into people with arthritis possibly. And obviously this could be a diagnostic for inflammatory disease in folks with Lyme disease. So we're pretty excited about moving that along. And again, you can get incredible architecture, and we're hoping to get some drilled down pathology in histo to see if we can actually image the invading immune cells in these regions of inflammation. So we have done some classic analysis of the PMCs from these mice. We can show our different molecules in inactive versus inactive. We can profile these different T cell populations and start to break them down. Most of the big signals are coming from CD4 positive and CD8 cells, we think. We can correlate that with the protein expression and start to break down, which is more interesting, the different types of immune cell responses we're seeing in that animal. So where we're excited, and this is happening with Monica, is to look at this same procedure in a mouse infected with Borrelia. So, and that is ongoing. So, and what do those T cell populations look like? And can we image it? And can we image it by PET? If we image by PET, we're going to clinical trials right away. So it would already be a great diagnostic test, actually, with these fluorescent probes alone to profile T cells in blood samples, as is, ready to go. Now, this is to end, and this is another probe, and it's one that came out of our screen that only specifically targets Borrelia HTPG, the Borrelia form of HSP90. It does not target the human form. And the way we start that and figure that out is we start biochemically, we express recombinant proteins, we test them, we do enzyme assays. This is what this noise is here, but actually to me it's like, wow, look at that. It's like really specific for that form. It really doesn't hit the E. coli version, but it definitely doesn't hit the human. So if we make that probe, and where we are with this, let's put this into first the cells, which is where we are, and then into the infection model. And if you could click that movie, this one, this is the biofilm. And if you look in 3D and you look at the probe, the red blobs are our probe. It's going into the biofilm. It recognizes the biofilm. My students call it, on these slides, we'll often see smears. They call it snot. And the probe is also recognizing the biofilm. So you could imagine in tissues in histology, you might see traces of that material, if not the aggregates. If you wouldn't mind clicking on this one, we look at different probes. This is NADA, which is a proteogliadine co-stain. And again, the green is the NADA, and the red is our probe. And then this one's very cool. This is a cluster of the spirochetes all together, all twisted together in 3D, and you can see these discrete red staining, which is our HS-198 probe in these little globs, and the green is CyPro, which is DNA. So you see the whole sort of organism. We're trying to get some TEM on this to get some more ultrastructure. But the point is that this probe is getting into the bug, and it's dragging a big old fluorophore. So that means we could drag other things in there and specifically target it. Will not target the human. So this is what we're really excited about, obviously. So, and then you can do wonderful things like go through the different, if you take a field, it's very difficult to synchronize these organisms in culture, but you can go through the fields, you see your biofilm aggregates. These are, the longer the culture it goes, the more you see. We do see this depth as well. You can see the little spirochetes have got, there's not a bright field, but a dark field image of an organism, and you can see how it co-stains with our organism. That's with our dye, it's the blue one. That's another type of dye that recognizes proteins within the organism. Oh, this is the snot. This is something that's laid on the surface that doesn't seem to have anything in it, but it seems to be like a smear. And again, another organism over here. So this is pretty cool stuff. So the goal of all of this is to try and develop a pet agent, I think, to look at people with active disease and see if we can't do two things. One is, do we see immune cell localization or flare-ups in regions where you've got pathology going on? But very coolly, could we see evidence of the organism itself? Because these pet agents that we use are exceedingly sensitive. They see not down to single molecules, but certainly down to single cells. And so I think that would be of some benefit if we can actually see within people's tissues sites of infection or where these little critters are hiding. So that's the hope. So, sort of I'll quickly go through. We're going things that we are doing. We are continuing with this large-scale program. We're hoping to develop or identify many leads, many chemical starting points. With the Cohens, we'll just dump this into the public domain somehow so that other people can pick them up. We can't do them all. We are really active. We are much closer to the clinic with this series of inhibitors, these HSP90 inhibitors that now potently inhibit the Borrelia version. And we're obviously, with Monica, now moving these into animal models, and that is ongoing as soon as IACACs are finished and signed. And we'll probably end up making a novel, more drug-like molecule. It's not just an imaging agent, but something that is antimicrobial, perhaps. And then I would really trying to develop the pet versions to do both inflammation and infection in vivo. Nobody's done that with any kind of disease, and that would be a big thing. And it will only take some convincing preclinical efficacy data or demonstration in models. First, a rodent model, and then with Monica, a primate model, I think to convince the FDA that you could move this into a clinical trial in patients, just to see if it works. So I don't think we're more than a year or two away from that, getting into that. Depends on how much money we can convince people to part with to do the IND campaign, which is really the only thing in our way right now. So these are the people that are involved in this work. Neil brought me into this. This is part of Neil's vision. I'm happy to be part of it. And where is Monica? I'm glad she's not here. She's not on my slide. That was a terrible episode, but this is a major person. If you'd been here yesterday, had big, glowy... But she was on the first one, right? So these are my guys, and they're all great. I have chemists and graduate students. And we are funded no longer by NIH anymore, but DoD, openphilanthropy.org. This is the Facebook initiative, the Cohen Foundation, We work with a number of CROs that help us do some of the animal work. But thank you.